Hello everyone. So here I have taken all the material required in this experiment. Here I have taken onion, water bottle, glycerin, and then here it is Safranin stain solution. After that, here you can see there are two watch glasses containing Safranin stain solution, and another one is having water. Then we'll be needing scissor, needle, brush, and to observe this slide we'll be requiring this compound microscope. So let's start it. Firstly, I'll peel off very thin transparent onion peel from the inner side using forceps. Here you can see this is the onion peel. I'll put it in the water so that it doesn't get dried. Now I am transferring the peel into safranin stain and will leave it for few minutes so that the cells get properly stained. Here I'll again transfer it into the water to remove the extra staining. Next I will take a glass slide and very carefully I'll be placing this onion peel on the glass slide using the brush. Here we need to remove this extra water and staining using blotting paper or filter paper. Next, here I am adding a drop of glycerin on the peel. This glycerin helps in preventing the slide from drying and maintains the natural shape of the cells. So at the end, I'll be using brush to spread it evenly. And again, I will remove the extra staining or glycerin. This will help us to prepare a clear slide. So here you can see the slide. Next, I will be placing a cover slip over it. So I'll be using forceps for this thing. We need to be very careful while placing it so that there is no bubble formed inside it. So here the slide is ready. Next, I will put this slide under the microscope. So here we need to adjust the light entering into it and also the power of the lens. So I'll be adjusting it so that we can clearly see the cells of onion peel. So finally, here you can see the onion peel cells. Can you identify the cell wall and the nucleus in each cell? And if this helped you in learning, please like, share and subscribe to my channel. Thank you for watching. Stay connected.